Welcome to Biology, the scientific study of life. Respirators in the plasma membrane. Third one is ion channel respirators. A ligand-gated ion channel is a type of membrane respirator, a region of which can act as a gate when the receptor changes shape, when a signal molecule binds as a ligand to the respirator protein. The gate opens or closes, allowing or blocking the flow of specific ions, such as in A positive or Ca2 positive, through a channel in the respirator. Like the other respirators we have discussed, these proteins bind the ligand and at a specific site on their extracellular side. Notice picture 1. Here we show a ligand-gated ion channel receptor in which the gate remains closed until a ligand binds to the receptor. Picture 2. When the ligand binds to the receptor and the gate opens, specific ions can follow through the channel and rapidly change the concentration of that particular ion inside the cell. This change may directly affect the activity of the cell in some way. Picture 3. When the ligand dissociates from this receptor, the gate closes and ions no longer enter the cell. Ligand-gated ion channels are very important in the nervous system. For example, the neurotransmitter molecule released at a synapse between two nervous cells. Notice that. Bind as a ligands to ion channels on the receiving cell, causing the channels to open. Ions follow in and trigger an electrical signal that propagates down the length of the receiving cell. Some gated ion channels are controlled by electrical signals instead of ligands. These voltage gated ion channels are also crucial to the functioning of the nervous system. Transduction cascades of molecular interactions relay signals from receptors to target molecules in the cell. When signal receptors are plasma membrane proteins, like most of those we have discussed, the transduction stage of cell signaling is usually multi-step pathway. One benefit of such pathways is the possibility of greatly amplifying a signal if some of the molecules in a pathway transmit the signal to multiple molecules of the next component or in the series. The result can be a large number of activated molecules at the end of the pathway. In other words, a small number of extracellular signal molecules can produce a large cellular response. Moreover, multi-step pathway provides more opportunities for coordination and regulation than similar systems do. Signal transduction pathways. The binding of a specific signal molecule to a receptor in the plasma membrane triggers the first step in the chain of molecular interactions, the signal transduction pathway that leads to a particular response within the cell. Like falling dominoes, the signal activated receptor activates another protein, which activates another molecule, and so on, until the protein that produces the final cellular response is activated. The molecule that relay a signal from receptor to response, which we call relay molecules in this book, are mostly proteins. The interaction of proteins is a major theme of cell signaling. Indeed, the proteins interaction is a unifying theme of all regulation at the cellular level. Keep in mind that the original signal molecule is not physically passed along a signaling pathway. In most cases, it never even enters the cell. When we say that the signal is relayed relayed along a pathway, we mean that certain information is passed on. At each step, the signal is transduced into a different form, commonly a conformational change in a protein. Very often, the conformational change is brought about phosphorylation. Protein phosphorylation and dephosphorylation. A previous chapter introduced the concept of activating of a protein by adding one or more phosphate groups to it. Notice in this video we have already seen how phosphorylation is involved in the activation of receptor tyrosine kinases. In fact, the phosphorylation and dephosphorylation of proteins is a widespread cellular mechanism for regulating protein activity. The general name for an enzyme that transfers transfers phosphate groups from HB to a protein is protein kinase. Recall that receptor tyrosine kinases phosphorylate other receptor tyrosine kinase monomers. Most cytoplasmic protein kinases, however, act on a protein different from themselves. Another distinction is that most cytoplasmic protein kinases phosphorylate either the amino acid serine or threonine rather than tyrosine. Such serine or threonine kinases are widely involved in signaling pathways in animals, plants, and fungi. Many of the relay molecules in a signal transduction pathways are protein kinases, and they often act on other protein kinases in the pathway. Depicts a hypothetical pathway containing 3 different protein kinases with create a phosphorylation cascade. The sequence shown is similar to many known pathways, including those triggered in yeast by mating factors and in animal cells by many growth factors. The signal is transmitted by a cascade of protein phosphorylations, each bringing with it a conformational change. Each shape change results from the interaction for the newly added phosphate groups with charged or polar amino acids. The addition of phosphate groups often changes a protein from an inactive form to an active form, although in other cases phosphorylation decreases the activity of the proteins. The importance of protein kinases can hardly be overstated. About 2% of our own genes are also to code for protein kinases. A single cell may have hundreds of different kinds, each specific for a different substrate protein. Together, they probably regulate a large proportion of the thousands of proteins in a cell. Among these are most of the proteins that in turn regulate as cell reproduction. Abnormal activity of such a kinase can cause abnormal cell growth and contribute to the development of cancer. Equally important in the phosphorylation cascade are the protein phosphatases, enzymes that can rapidly remove phosphate groups from proteins, a process called phosphorylation by dephosphorylating and thus inactivating protein kinases. Phosphates provide the mechanism for turning off the signal transduction pathway when the initial signal is no longer present. Phosphatases also make the protein kinases available for reuse, enabling the cell to respond again to an extracellular signal. At any given moment, the activity of a protein regulated by phosphorylation depends on the balance of in the cell between active kinase molecule and active phosphatase molecules. The phosphorylation or dephosphorylation system acts as a molecular switch in the cell, turning activities on or off as required. Small molecules and ions as second messenger. Not all components of signal transduction pathways are proteins. Many signaling pathways also involve small non-protein where water-soluble molecules or ions called second messengers. Receptor the extracellular signal molecule that binds to the membrane receptor is a pathway's first messenger. Because second receptor is a pathway's first messenger, because second messengers are both small and water soluble, they can readily spread throughout the cell by diffusion. For example, as we will see shortly, it is a second messenger called cyclic AMP that carries the signal initiated by epinephrine from the plasma membrane of a liver or muscle cell into the cell's interior, where it brings about glycogen breakdown. Second messenger participates in pathways initiated by both G protein linked receptors and receptor tyrosine kinases. The two most widely used second messengers are cyclic AMP and calcium ions Ca2 positive. A large variety of relay proteins are sensitive to the cytosolic concentration of one or the other of these second messengers. Cyclic AMP, once Errol Sutherland had established that epinephrine somehow causes glycogen breakdown without passing through the plasma membrane. The search began for the second messenger. He coined the term that transmits the signal form of plasma membrane to the metabolic machinery in the cytoplasm. Sutherland found that the binding of epinephrine to the plasma membrane of a liver cell elevates the cytosolic concentration of a compound called cyclic adenosine monophosphate, abbreviated cyclic AMP or C-small AMP. An enzyme embedded in the plasma membrane, adenylyl cyclase, converts ATP to a C-small AMP in response to an extracellular signal, in this case epinephrine, but the epinephrine doesn't stimulate the adenylyl cyclic directly. When epinephrine outside the cell binds to a specific receptor protein, the protein activates adenylyl cyclase, which in turn can catalyze the synthesis of many molecules of C-small AMP. In this way, the normal cellular concentration of a C-small AMP can be boosted 20-fold in a matter of seconds. Bon. The C-small AMP broadcasts the signal to the cytoplasm. It doesn't persist for long in the absence of the hormone because another enzyme called phosphodiesterase converts the C-small AMP to AMP. Another surge of epinephrine is needed to boost the cytosolic concentration of C-small AMP again. Subsequent research has revealed that epinephrine is only one of many hormones and other signal molecules that trigger the formation of C-small AMP. It has also brought to light the other components of C-small AMP pathways, including G protein linked receptors, and protein kinases. The immediate effect of C-small AMP is usually the activation of serine or threonine kinase called protein kinase A. The activated kinase then phosphorylates various other proteins depending on the cell type. The complete pathway for epinephrine stimulation of glycogen breakdown is shown here. Further regulation of cell Metabolism is provided by other G protein systems that inhibit adenylyl cycles. In these systems, a different signal molecule activates a different receptor, which activates an inhibitory G protein. Now that we know about the role of C-small-a and P in G protein signaling pathways, we can explain in molecular detail how certain microbes cause disease. Consider cholera, a disease that is frequently epidemic in places where the water supply is contaminated with human feces. People acquire the cholera bacterium Vibrio cholerae by drinking contaminated water. The bacteria colonize the lining of the small intestine and produce a toxin. The cholera toxin is an enzyme that chemically modifies a G protein involved in regulating salt and water secretion. Because the modified G protein is unable to hydrolyze GTP to GDP, it remains stuck in its active form, continuously stimulating adenylyl cycles. To make C-small-a and P, the resulting high concentration of C and P causes the intestinal cells to secrete large amount of water and salts into the intestines. An infected person quickly develops profuse diarrhea, and if left untreated can soon die from the loss of water and salts. Our understanding of signaling pathways involving cyclic A and P or related messengers has allowed us to develop treatments for certain conditions in humans. When such a pathway uses cyclic GMP or C-small-GMP as a signaling molecule, its effects including relaxation of smooth muscle cells in artery walls, a compound that inhibits the hydrolysis of CGMP or GMP, thus prolonging the signal. Was originally prescribed for chest pains because it increased blood flow in the heart muscle under the trade name Vigra. This compound is now widely used as a treatment for erectile dysfunction. Viagra causes dilatation of blood vessels, which allows increased blood flow to the penis, optimizing physiological conditions for penile erections. Okay, thanks for listening.